Hello, everyone. My name is Tesleem Lawal, and I will be presenting this poster on aqueous nanobubble dispersion for geological carbon storage. So I would first like to acknowledge my colleagues who also worked on this project, Hao Wang, Sophia Nacho, and our PI, Dr. Ryosuke Okuno. So for a bit of background, large-scale CCS technologies have gained significant attention as a means to mitigate greenhouse gas emissions and also to address global warming. And so one promising strategy for CCS is the storage of gas in water due to its potential for secure and permanent storage of gas in the subsurface. For example, carbonated water injection has gained attention for CO2 in situ mineralization in geological carbon sequestration. However, one obvious limitation of this carbonated water injection is that its CO2 concentration is limited by its inherent solubility. And so one promising method that has been studied for increasing the amount of gas in water is by dispersing that gas in the water in the form of nanobubbles. And so this figure here is a photo of an aqueous fluid, which is water, containing some nanobubble dispersions of nitrogen. And so because the bubbles are so small, submicron sized, the water appears to be transparent. And so nanobubble dispersions have been widely studied for open systems near atmospheric pressure. And however, their fundamental properties are not well understood, especially at high pressures, which of course are relevant to our subsurface applications. And it makes sense that they're not well understood at high pressures because it's not easy to directly measure these properties at high pressures. And so the main objective of our work is to investigate the effectiveness of gas nanobubble dispersions for geological carbon storage. And for that, we will develop models for the physical and transport properties of aqueous nanobubble fluids at a wide range of expected operating conditions. And to do that, we have to first determine the fundamental physical and transport properties of the aqueous nanobubble fluids. So for this work, we studied both nitrogen and CO2 nanobubble dispersions to understand the behavior of different gas species in aqueous nanobubble dispersions. For our methodology, we developed a program to measure the gas contents of nitrogen and CO2 nanobubble dispersions at pressures up to 4,000 psia, and we also analyzed the experimental data using a thermodynamic model. And so this figure shows the experimental setup for generating the aqueous nanobubble dispersions. And so we did it at different operating conditions. So pressures, injection rates, co-injection ratios, salinities, and pH. So for this, we had two accumulators, one containing either nitrogen and CO2, or the other one containing either DI water and NaCl brine. And we injected them through porous membranes that are housed in a core holder, and we collected the nanobubble fluid inside a receiver accumulator downstream of the core holder. And so the average pore size of this membrane was about 5 microns. And what we hypothesize is the reason for the nanobubble generation is that at first we have microbubbles that form from the gas snap-off that's happening across the interface of the membranes. And then at our elevated pressures, the bulk microbubbles are compressed into bulk nanobubbles. And so after generating these bulk nanobubbles, we then measure the gas contents. And so to do that, we first transfer the nanobubble sample into a sapphire cell. And so the purpose of using a sapphire cell is that a sapphire cell can withstand really high pressures and also high temperatures. And we can also visually monitor the behavior of the nanobubble sample. And so after transferring the sample into a sapphire cell, we connected it to a piston accumulator and ensured that the entire system was closed. And then we depressurized the nanobubble sample to atmospheric pressure. And so this depressurization process pushes the piston of the accumulator down. That in turn displaces some water from the accumulator. And so the displaced water corresponds to the amount of nitrogen that was present in the sapphire cell or in the nanobubble sample at atmospheric pressure. And then we also measure the amount of water that's remaining in the sapphire cell at atmospheric pressure. And we calculate all of this to determine the in situ amount of gas that was present in the nanobubble sample at the experimental pressure. And so after the experiments, we used a thermodynamic model to analyze the experimental data. And the benefit of the thermodynamic model is that it can give us an internally consistent set of properties for the experimental conditions, especially unmeasurable properties in high pressure experiments, such as the phase compositions and capillary pressure. And so for the thermodynamic model, we use the Gerg 2008 equation of states. And we also modeled it as a thermodynamically stable state of a closed system. And our model is able to estimate properties such as the bubble size, the bubble number density, as well as the capillary pressure of the system. And so I will be presenting the results for both nitrogen and CO2 nanobubble dispersions. And so for nitrogen dispersions, we see that as the pressure increased, the gas content also increased significantly. And overall, the solubility enhancement factor was about 3 times. And so at these high pressures, the gas content was 3 times higher than the solubility of nitrogen in water at the same pressure. And one other thing we did was we also tried to do different co-injection ratios and also different total injection flow rates. And we saw that those had a negligible impact on the gas content for nitrogen. Also for CO2, we see that as the pressure increased, the gas content also increased. However, it reached a plateau at around 2000 psi, and the final solubility enhancement factor was 1.5. And similarly for the nitrogen case, changing the co-injection ratio or the total injection flow rates had a negligible impact on the gas content. And then applying the thermodynamic analysis to the experimental data, for the nitrogen case, we see that actually for both the nitrogen case and the CO2 case, we see that the diameter decreased and the number density of the bubbles increased as the external water pressure increased. And we also see that a large fraction of the gas in the system is molecularly dissolved in the external aqueous phase. And so essentially, the existence of bubbles increases the gas contents in the system by increasing the supersaturation, which is basically the molecular dispersion in the aqueous phase. And so the presence of bubbles just tends to increase the level of supersaturation with capillary pressure, as shown here. And so what these results also show is that supersaturation is the main contribution to the amount of gas present in this aqueous nanobubble fluid. And that's why we have an increased gas content. And this finding actually has a lot of fundamental impacts on the research and development of nanobubble technologies. And so in conclusion, we developed a novel experimental program to generate aqueous gas nanobubbles at elevated pressures. We also measure the gas contents in the nanobubble sample. And so as the pressure increased, the gas content increased for both nitrogen and CO2, and their solubility enhancement factors range from 1.5 to 3 times the solubility limit. And we also developed a thermodynamic model to enable us have a fundamental understanding of various factors affecting the properties of aqueous nanobubble fluids. And so one other thing we did was we tried to depressurize the system from atmospheric pressure and then visualize what is going on in the nanobubble sample. And so if you scan this QR code, you see a video of the nanobubbles at atmospheric pressure. And so here is the video. And so we used nanoparticle tracking analysis to visualize the behavior of the CO2 nanobubbles at atmospheric pressure. And we showed that we had about greater than 10 to the 9 bubbles per milliliters at atmospheric pressure. And so we say greater because this was actually the equipment we used had a limit of about 10 to the 8, 10 to the 9. And so it's possible then that we actually have more bubbles in this sample at atmospheric pressure. And so that concludes my presentation. Thank you so very much for your attention.